So, in the reception phase of a G protein, um, they are the extracellular side on the cell membrane. So, when the ligand binds to this extracellular side of the G protein in the cell membrane, the receptor changes shape and is begun to activate, and thus the cytoplasmic side of the receptor then binds to the inactive G protein. After this, the signaling of the molecule on the G protein then creates chemicals and the GPCR, which is the gene protein coupled receptor on the cell membrane, to uh, go on to the next phase of the cycle, which is transduction.
For the step of transduction, the cytoplasmic side of the receptor, or the side that is inside the cell, binds to an inactive G protein. When the G protein exchanges GDP for GTP, the G protein activates. In this process, the G protein dissociates from the receptor by separating the alpha subunit from the beta and gamma subunits. Then the G protein, which is depicted as the active alpha subunit and GTP, diffuses along the cell membrane and binds to an enzyme. When the enzyme changes shape and activates, it leads to a cellular response. The cellular response acts as an effect of an on switch or an off switch that either causes or stops a cell function. Specific responses that are associated with G proteins and G protein coupled receptors are the human senses of vision, smell, and taste. Also, as it was shown in Hey Now Science's video of signal transduction pathway examples, in the fight or flight response, the G protein pathway can cause a cellular response to produce glucose for cellular respiration and body movement. In this example, when the adenylyl cyclase enzyme activates, it uses ATP to produce the second messenger of cyclic AMP, which produces protein kinase A. This causes a phosphorylation cascade in which the protein kinase A would create protein kinase 1, which phosphorylates protein kinase 2. Finally, protein kinase 2 would break down glycogen into glucose that could be used for cellular respiration for increased movement in the fight or flight response. Due to the G protein pathway reaction being temporary, after the cellular response, GTP is hydrolyzed into GDP and the signaling molecule unbinds from the receptor. The dissociation G protein subunits of the alpha, beta, and gamma subunits reunite for the next G protein pathway.